Good morning, students. I am Dr. Abhishek Kumar Singh, Tutor, Department of Physiology, NMCH Gaya. And today is a continuation of the last class which we were talking about, the central nervous system. And in that, we'll just revise the topics which we covered in the last class. We talked about the functional organization of the central nervous system, which included forebrain, midbrain and hindbrain, and their parts. Then we talked about the way the CNS function, that is the integrated function of the CNS. And then we talked about type of cells in CNS, which included two types of cells, the glial cells and the neurons. We talked about the glial cells in the last class, and today we are going to talk about neuron. So as we know, neuron is the functional unit of nervous system, and it consists of three parts. Here we have focused a diagram which you can see clearly. The diagram which shows it consists of soma, which is called as the cell body, then it consists of dendrites, and it consists of axon. What is soma? Soma basically is the cell body, which includes nucleus and the cytoplasm. The nucleus contains a centrally placed or eccentrically placed nucleolus. The cytoplasm of the soma, that is the cell body of the neuron, contains endoplasmic reticulum, mitochondria, a prominent Golgi apparatus, cytoskeletal elements that are called as neurofilaments, which includes microtubules and microfilaments. Then we also have specialized granules which are present in the soma of neurons. As you can see in the picture, the dark dots which you can see in the picture consist of those specialized dots. I'll just show you by finger pointing. So these are the specialized granules, and these granules which are present in the soma are called as nasal granules or the nasal bodies. They are what? They are basically the modified rough endoplasmic reticulum. They act as a biosynthetic apparatus for synthesis of proteins in the neurons. So they are basically the rough endoplasmic reticulum, and they help in synthesis of proteins in the neurons. They are present throughout the soma except in the axon hillock. We'll talk about axon hillock in the coming topics. Then after soma we have the second structure. You can see the stem branch-like structures which are emerging from the soma of the cell body. They are called as dendrites. Dendrites occupy almost 90% of the total surface area of the neuron. They are what? They are which are the tapering processes of variable complexities that arises from the soma. As you can clearly see in the picture, the branch-like pattern which is coming out from the cell body or the soma of the neuron are the dendrites. The part which is very close to soma, as we also know in anatomy, the part which is close is called as the proximal dendrites, and the part which is away are called as the distal dendrites. There is a difference between the proximal dendrites and the distal dendrite. The proximal dendrites contains nasal granules and Golgi apparatus, but the part which is away, that is the distal dendrites, do not contain nasal granules. I will show you by indicating it on the picture that the proximal part, these proximal parts contains nasal granules, while when you go to the distal dendrites, you can clearly see, you can zoom it on your mobile or laptop which you are using. You can zoom it and you can see that the proximal branches of the dendrites do not contain nasal granules. This is one difference. Then they contain what? These dendrites contains microfilaments and microtubules. Along with that, the cell body, they are the main receiving end of the information, and they form the receptor zone of neuron. These dendrites are the receptor zone of neuron. Dendrites conduct impulses towards cell body. We have to know this. Dendrites conduct impulses towards the cell body. Then we come to another major portion of neuron, the part of neuron that is axon. Axons are what? Axons are the structure arising from the soma at a specialized region. You can see a long tail-like thing which is coming from the neuron is called axon. What is the difference between the axon hillock and what is the axon hillock? As you can see in the picture, I'll just show you another picture which will show this. Just show you. You can see here, this is axon hillock, which you can see that is the tapering part which is arising from the neuron. As you can see, the arrow is the axon hillock. This is the axon hillock, the tapering end. The difference between axon hillock, which forms the proximal part of the axon, and the proximal dendrites, which we have already studied before, is that axon hillock do not contain nasal bodies or the nasal granules. Nasal granules are not present in the proximal part of axon, which is called axon hillock. The area, the part which is just attached with the soma of the neuron. So this is the major difference, that axon hillock do not contain nasal granules, neither endoplasmic reticulum nor Golgi apparatus. Axon hillock continues as unmyelinated proximal part of axon, and they are called as initial segment. So axon hillock, which is unmyelinated, is called as initial segment. And what are these initial segments specialized for? They act as a spike initiation zone of neurons. We'll talk about all this when we talk about synaptic transmission. Spike initiation zone. These proximal part of neuron, which is axon hillock, are called as spike initiation zone of neurons, where action potential which arises here are due to the summation of the electrical activities that have occurred in the cell body. When various electrical activities which arises from the cell body, they undergo summation, then they result in an action potential. So this is the spike initiation zone. Then interior of axon. This is the interior of the axon. This axon is called, interior of the axon is called axon cylinder, which contains exoplasm. It is an exoplasm which is present in the interior of the axon, and these exoplasm contains mitochondria and neurofibrils as well as vesicles, but no nasal bodies. So these exoplasm, that is the interior of the axon, which consists of axon cylinder, and axon cylinder contains exoplasm, do not have nasal body, but they have neurofibrils. That is the cytoskeleton of the cell. So they have neurofibrils, vesicles, mitochondria, but no nasal bodies. Neurotransmitters which are synthesized. We'll talk about neurotransmitters in detail when we'll talk about in further classes. Neurotransmitters synthesized in the soma and are transported in the exoplasm by the means of axonal flow to the synapse. So the neurotransmitter are transported in the exoplasm from the cell bodies, that is soma. Then we come to, there is something which is, you can see here is myelin sheath. These myelin sheaths contains a type of a glial cells which are called as Schwann cells. You can see the picture of the Schwann cell. Schwann cell sheath around an axon. They form the myelination of the nerve fibers. We already know that nerve fibers can be myelinated as well as non-myelinated. So the myelinated nerve fibers will have the Schwann cell sheath around the axon, and they will cause myelination, that is electrical insulation of the axon. So the cells of the myelin sheaths are called as Schwann cells. You can clearly see here the Schwann cells being shown, and then Schwann cell nucleus being shown, and then the myelin sheath. Then in between these myelin sheath, these sheaths, there is constricted area which is known as node of Randall, which are the gaps between the myelinated nerve fibers. They act as an insulation. They act as an insulation areas of Schwann cells. So these are called as nodes of Randall. Then we come to classification or the types of neurons. I'll just show you the picture. I hope this is clear. So we have types of neurons can be classified or can be typed according to the number of processes or axonal arrangements. It can also be typed based upon the length of the axon. It can also be classified or typed based on their functions. And the fourth way to classify or type them is by their pattern of dendrites. So based on the axonal arrangement, one type of classification based on the processes, the one which you can see in the picture right here. So there can be unipolar. This is the unipolar one. Unipolar are the one soma consisting of a single process, which are found in invertebrates, and they are also found in vertebrates like in humans in autonomic nervous system. They are also present in touch and pain sensory neurons. They are of unipolar types. You can get MCQs with the type of neurons and like what type of neuron is a touch or a pain neuron. So it is a unipolar type of neuron. So you can have an MCQs on that. Then the second way of classifying, the second type, just like unipolar, there can be something called as bipolar neuron. You can see the picture of a bipolar neuron. A single soma with two axons going on. So two processes arise from the cell body of a bipolar neuron, as the name suggests. So example of a bipolar neuron is bipolar cells of retina, which you are already studying right now in a eye topic. So bipolar cells of retina, and also the olfactory neuron, the special senses which you have been studying. You will have the examples of the neurons are from that, the bipolar cells of retina and the olfactory neurons. They are the examples of bipolar neurons. Then we have multipolar neuron. These are the multipolar neurons. So multipolar neurons means many processes arises from the cell body. Examples of multipolar neurons are spinal motor neurons. They are the example of multipolar neurons. Other examples are like pyramidal neurons. They are also the examples of multipolar neurons. Then there is one more type which are known as anaxonic. You can see here, there is no axon. Anaxonic neuron. Anaxonic neurons are found in invertebrates just like the unipolar ones, but they are also seen in humans where they are seen as amacrine cells, which are the interneurons in the retina. So the amacrine cells are what? They are the interneurons in the retina, and they are also present in the retina horizontal cells. So these retina horizontal cells and the amacrine cells are the examples of an axonic neuron. A neuron which has a soma but doesn't have an axon. Then there is another classification which is not seen in the picture here, and that is called as a pseudo-unipolar neuron. This is the fifth type, fourth type, pseudo-unipolar. Here what happens, the axon split into a central and a peripheral processes. An example to that is a dorsal root ganglia. You can see here, the dorsal root ganglia has a central and a peripheral processes. So this is an example of a pseudo-unipolar neuron. So this is one way of typing or classifying the neuron. Then we come to the second way of classifying, that is by the length of the axon. There can be two types. One is called as Golgi type 1 and Golgi type 2. Golgi type 1 neurons are neurons with short axons and dendrites, and they terminate very near to the soma. Example are the cortical inhibitory neurons. We will know all about this when we go in details of central nervous system. So Golgi type 1 have very short axon and dendrites, and they terminate very near to the soma. They are not long like one which you have seen in the picture. Then there is Golgi type 2, which have long axons. Examples are the corticospinal tract. We will talk about this later. So cortical motor neurons, they have long axons. So based on the size, length of the axon, we have Golgi type 1, Golgi type 2. Then based on functions, there can be two types, sensory and motor. Sensory are those which carry impulses from receptors to the CNS, and they are known as afferent fibers. Then motor are those which carry from the central nervous system to the target organs, and they are known as efferent fibers or efferent neurons. Then the classifying neurons based on the pattern of dendrites. I have a picture for that. This. The classifying it based on the pattern of dendrite. You see the patterns of the dendrite. They are so beautifully looking dendrites. So what are these patterns like? They can be pyramidal cells type and stellate cells type. Pyramidal cells are, these are pyramidal cells. You can see here. Pyramids. Pyramids are those which spread like pyramids. They spread like pyramids. Example to that is a hippocampal pyramidal neurons. And then is stellate cells. They have a radial shape spread of dendrites which occur in cells like cortical stellate cells. So the stellate ones have a radial shape spread, while the pyramidal type, which you can see here, they have a spread like pyramids. Okay. Then we also have the what we have talked, the bipolar neurons, the retinal neurons, and the olfactory neurons, and then unipolar neurons. You can see here the touch and the pain sensory neurons we have talked about, and the anaxonic neurons. You can see here there's the soma is there, and there is no axon, and this is the amacrine cells which is present in the retinal layer. So this was all about the type, the second type of cell in the central nervous system, that is neuron, their types, their structure, and details. Then we start with a new topic today itself, and we continue that in the coming lectures. And the topic is synaptic transmission in the central nervous system. What do you mean by synapse? Synapse basically, the word means connection. So what is synaptic transmission? It is a communication in the CNS which is achieved through synapse. That is the communication which is achieved through these synapses. Each neuron forms about 2000 synapses, and there is about 10 to the power of 11 neurons in CNS. So how many synapses we can have? It will be around 2 into 10 to the power of 14. 2000 synapses, 10 to the power of 11 neurons, and then so the total number of synapses will be 2 into 10 to the power of 14 number of synapses. Now classifying the classification of synapse. Synapse can be classified according to the part of neuron which is involved in formation of synapse. I have a picture for that, figure for that. I'll show you. So this is the classification based on, see, this is the synapse. There is one neuron, cell body, there is dendrites, and then there is synapse happening. That is the connection happening between the two neurons. So based upon the part of the neuron which is involved in the synapse, there are 4 types in CNS. First and the most commonest type is called as exodendritic synapse. They are the most common. Around 80% in cerebral cortex, the synapse are of exodendritic synapse. As the name suggests, it's very easy to make out. Axon of presynaptic neuron connects with the dendrite of postsynaptic neuron. So axon of one presynaptic with the dendrites of postsynaptic. We'll talk about presynaptic and postsynaptic in details when we talk about synapse, more in details then. So here the axon of one, we'll see the axon of one is connecting with the dendrite of the another. So it is exodendritic synapse, and they are the commonest one. You can get an MCQ on this. So So these are exodendritic synapses, and they are the commonest one in cerebral cortex. About 80% of the synapses in CNS are of exodendritic type. Then second one is exosomatic. You can see here exosomatic. Here is exosomatic. Exosomatic, as the name suggests, axon of presynaptic neurons with the soma, that is the cell body of the postsynaptic neuron. They forms a synapse, and this is known as exosomatic synapse. They are again a common ones which are found in the CNS. Around 15% of the synapse forms are exosomatic type. Then two others are exonic. As the name suggests, axon of presynaptic with the axon of postsynaptic. Axon of one with the axon of another. This is exonic. Axon of one with the axon of another. Exonic. Okay? And then comes the fourth type, that is dendrodendritic, which are very rare, are dendrodendritic. Name suggests dendrites of one. This is not shown here. The dendrite of one with the dendrite of another, that is dendrite of presynaptic with the dendrite of postsynaptic, and this is a very rare type of synapse. Then the classification of synapse based upon the nature of transmission. It can be chemical, it can be electrical, it can be conjoined. Chemical are the one which are the most common in CNS, that is the transmission through the release of neurotransmitters. Then comes the electrical, that is through the gap junctions, rare in CNS. And then is the conjoined, means there is partly electrical and partly chemical. Then third way of classifying the synapse can be according to the number of neurons involved. It can be one-to-one, like one neuron ends on another, as we can see here. One neuron is ending on another. So, example is neuromuscular junction, and it can also be seen in parasympathetic system. Then it can be many to one, multiple neuron terminating on a single neuron, and it is very commonly found in CNS, that is in sympathetic nervous system, and one to many, which is a rare type where one neuron contacts many neurons. So rare type is one to many, one neuron contacts many neurons. Common ones are many to one, which is more common in CNS, that is multiple neurons terminate on a single neuron. Then mechanism of synaptic transmission, in which we will talk about in the next class, where we will talk about the functional anatomy of the synapse, and we will talk about steps of synaptic transmission.